Hello, this is Vijay Singh. I am Associate Professor and Head in the Department of the Bioscience, Indrasil University, Mehsana. So I would like to talk on the DNA repair, which is important mechanism in the molecular biology, as I have been trained as a genetic engineer where I have clone, express and characterize the recombinant protein in the E. coli bacterial system. Then I have been trained as a postdoc in the synthetic biology, metabolic engineering and the genome engineering, where I learn a lot of new techniques that can be used in the molecular biology. So as we know, the DNA repair, it is a very important mechanism in the bacterial cell, and it is going to happen, and the DNA is going to be damaged through the different way. The first is the reactive oxygen species, that is called the ROS. The second is the replication error, where the DNA polymerase is going to cause the error during the DNA replication and the basis is going to change. The third one is the X-ray. If you put the X-ray on the DNA is going to damage. UV light is the another source of the DNA damage. There is the alkylating agent that causes the DNA damage. And sometimes there is the problem with the spontaneous reaction that is caused the mutation. So during the DNA damage, the DNA is going to affect in the several way. That's the DNA is going to be repaired somehow. So the cell have the own mechanism that can repair the damaged DNA through the limited synthesis with the small fragment. And if the DNA is not going to damage, they are going to cause the cell cycle arrest. In that way, the apoptosis is going to happen, mutation is going to happen that can lead to the cancer, and there are the number of genetic disorder. So this is the effect of when the DNA is going to damage. So this DNA damage is going to be repaired in the several way. The first, the DNA damage is going to happen with the ionizing radiation, X-ray and the anti-tumor agent. That is going to cause the double stranded break or single stranded break or intra-cross link or inter-cross links. So in intra-cross link means there is the internal one strand base pair they are going to cross link, and inter strand cross links mean the both the anti-parallel strand they are going to cross link. So that is the inter and the intra strand cross link, and this cross link is going to repair by the non-homologous end joining. And in the non-homologous end joining, there is the random joining of the breakage DNA, and this is going to be happen normal in the cell, and the fidelity means the joining ability is little less and is less frequently found. Then another mechanism for the DNA joining in the homologous recombination mechanism. So in the homologous recombination, there is the both the end 3' and 5' they have the homologous region, and this DNA is going to recombine with where the DNA damage is going to happen, and this is going to be more fidelity. So this is the quite dominant mechanism found in the mammalian system. And the second, the DNA damage is going to happen with the UV light. So if you put the UV light, we expose, organism exposed with the UV light, certain chemicals they are going to be make and the DNA damage. So when the DNA is going to expose with the UV light, the thymine dimer it is going to form, and there are the bulky ducts and the pyrimidine dimer is going to form, and this pyrimidine dimer is going to be repaired by the nucleotide excision repair mechanism, and this is the quite dominant pathways for the repairing of the DNA. The third category of the DNA damage is oxygen radicals, hydrolysis, alkylating agent. So this is the A basic site where the single stranded DNA is going to be break, and the lesion is created by the 8-oxoguanine lesion in the DNA, and this is also going to repair by the base pair excision repair mechanism, and this is also very dominant pathways in the repair. And the last category of the DNA damage is going to happen with the replication error. So in the DNA during the replication, they are going to form the error. And this error is going to create the mismatch bases. Either insertion is going to happen, yeah, either the deletion is going to happen. And this repair is going to repair by the pathway that is called the mismatch repair mechanism, that is the MMR. And this pathway is quite strong in the system. So in the mismatch system, if the DNA error is going to happen, this mismatch repair system is going to correct the DNA. And why DNA repair is the necessary? Because when the DNA is damaged, if you see, it is very important to repair to make the and maintain the genetic stability of the DNA. Otherwise, if the genotype is changed, then there is the problem, the phenotype is going to change. And there is the defective incomplete DNA repair. Sometime it is going to happen. That's lead to the genetic instability. So if the DNA is not enough stable and they are going to have the mutation inside, there is the issues, there is the cause, there is possibilities of the causes, the hereditary disease, cancer, and another is the genetic divergence. So it is going to create, so that's why the DNA repair is quite important and essential for the repairing of the DNA once they are going to be damaged. So the DNA repair inside the cell, there are the two way of the DNA is going to happen. So one is the single strand break, it is going to happen, and the second is the double stranded break. So these, if one DNA single strand is going to be damaged or both the DNA is going to happen, they have the different pathways that can help to make the DNA repairing. So if the single strand DNA damage is going to happen, then methyl directed mismatch repair, that's help to make the DNA repairing. Second one, the base excision repair. Third one is the nucleotide excision repair. Fourth is the direct repair by the methyl transferase alpha ketoglutarate. They can help to make the single stranded DNA damage correction. And the second is the double stranded break, which is going to happen, and it is going to repair by the mechanism where non-homologous end joining, it is help to make the DNA repair. So only issue in the non-homologous end joining, sometimes the base pair is going to be added inside the gene, yeah, base pair is going to remove from the gene. So sometimes this is the like making the knockout of the gene, yeah, knock in of the gene. So sometimes the function can be restored, yeah, function can be deleted, yeah, function can be dysfunction. So this way how the non-homologous end joining pathway is working. And the second one is the homologous repair recombination. So where it's require the homologous region of the both side of the DNA strand, then they can recombine with the lambda ray system. So in the E. coli, so DNA replication is going to happen when the GATC, this is the region where this is the palindromic sequences where the methylation is going to happen. So there is the enzyme that is called the deoxyadenosine methylase, that is called the DIAM. They are going to add methyl group to the adenine side. So when both the DNA, the adenine is methylated, the DNA replication it is going to happen. When the DNA replication is going to happen, they make one strand is the methylated and the daughter strand is non-methylated. So you can easily identify if in methylated DNA or non-methylated DNA. So methylated DNA is belongs to the parents and the non-methylated belongs to the daughter cell. Until the new strand or non-methylated strand are now undistinguishable. So once both the strand is the methylated, then the second level of the DNA replication it is going to happen. So the methylation is the important before starting the DNA replication. So this is the one important steps for the methylation. So in the methyl directed mismatch repair, where the DNA polymerase, it is going to move in the replication fork of the DNA replication. So if there is a mismatch between this bases where they are going to replicate, the polymerase 3, DNA polymerase 3, this is an enzyme that can incorporate sometime 10 to power 6 one base pair in 10 to power 6. There is the issues, there is the mismatch, it is going to happen. So DNA polymerase 3 have the proofreading property, so they can repair themselves. And the actual mutation rate is 1 in 10 to power 9. So 10 to power, they are 10 to power 10. So this is the mutation rate found in normal during the replication. And if you see, there is the methyl mismatch repair system. So this is the E. coli system where methyl DNA directed DNA mismatch repair system is going to active. They can remove and replace the mismatch base pair. So adenine is in palindromic GATC sequences is methylated by the time. So enzyme that can recognize the parental strand by looking the methylation parent, and the rotary strand, it is not methylated. So if you see, there is the enzyme that can help to make this repair system. So that's called a series of enzymes, that's mut S, mut L, and mut H, that act to repair the mismatch system. So mut S find first the mismatch where it is going to happen. They can recruit mut L and mut H enzyme. Mut L recognize the methylated strand and the brings in a loop. Mut H then cleave the unmethylated strand containing the mutation near the GATC. So they are going to bind on this mismatch site, and first the mut S is going to recognize and bind with this nearby the mutation site, and the mut L they are going to recruit may act like the molecular matchmaker. So that can promote the formation of a stable complex between the two or more DNA binding protein. And this process is ATP dependent process. So they are going to recognize the methyl strand and recognize the mut S. Mut H is the another protein that is sequence specific endonuclease. They can form the loop, cleave the unmethylated DNA GATC sequences. And once they make this cut or nick in this GATC sequences nearby, then there is another enzyme called the UBRD that is have the helicase activity. So this UBRD that have the helicase property, and they can unwind the cleavage strand, exposing in to a variety of exonuclease. The result is the gap is going to fill by the DNA polymerase by the DNA synthesis. And there is another enzyme, they are going to help the XO1, XO7, 8, XO10, and the RAGZ. They are going to help to remove the base from the daughter strand. And the polymerase 1, they are going to involve in the DNA replication or DNA synthesis, and the gap is filled by the DNA polymerase 1. So this DNA polymerase, they are making the synthesis of the DNA, and they can goes until the where the damage is going to be repaired. So DNA polymerase pass through the region where mismatch exists. They can insert the correct base pair, and after the sometime DNA polymerase fall off from the DNA strand, and with the help of the DNA ligase, that is the enzyme, they can seal the gap between the two DNAs. So this enzyme is sealing the nick of the replicated DNA. So in this way, the DNA repair, it is going to happen. So the second single stranded DNA break, it is going to repair, that is called the base excision repair. So in the base excision repair, and we know there is the only four nucleotide, that is the adenine, guanine, cytosine, and thymine, it's available and make the DNA. And this is going to happen with this deamination of the cytosine. So cytosine, it is going to convert into the uracil, That's the deamination of the cytosol. So amino group is going to remove, and this phosphate backbone in the nucleotide, and here is the cytosine, then phosphate backbone. So the guanine and the cytosine, they are going here in another strand. This is the adenine and the uracil. They are going to form the bond between this adenine and uracil, and the guanine and cytosine bond, the triple bond, hydrogen bond with the G and C. So deamination of the adenine, where the adenine is going to deaminate and produce the hypoxanthine molecule. Once they are going to methylate this adenine, they make the 3-methyl adenine. So the adenine, hypoxanthine, and 3-methyl adenine. And there is the enzyme and hypoxanthine deamination of the adenine, and the 3-methyl adenine, this is the methylation of the adenine. So the adenine is going to convert whether through the hypoxanthine by the deamination process, or through the methylation process they are going to make the 3-end methyl adenine. And this is going to make like thymine, cytosine, and you can normally see how they are going to be changed and how they are going to substitute. So the recognition enzyme for this hypoxanthine, 3-methyladenine, and the uracil, and they are going to make the cleavage. So hypoxanthine DNA glycosylase, this is the enzyme. This hypoxanthine molecule, it is going to be deaminated. They are going to hydrolyze this deoxyribose and glycosidic bond, and they can excise the hypoxanthine, a deamination product of the adenine, from the double-stranded DNA. And they lack any detectable uracil DNA glycosylate activity. And another conversion, methylation of the adenine, that's make the 3-methyl adenine. So there is the enzyme that is called the 3-methyl adenine DNA glycosylase, and this enzyme make this 3-methyl adenine through the methylation of the adenine. They make the hydrolysis of the 3 hydrox hydrolysis of the 3-deoxyribose and glycosidic bond. They can excise 3-methylene from this damaged DNA. And the uracil DNA glycosylase, this is the product of the when the cytosine is going to make the deamination process and make the uracil. They are going to hydrolysis through the deoxyribose and glycosidic bond and excise this uracil residue from the DNA. So in this way, uracil is going to remove from the DNA molecule. And normal, there is the oxygen, reactive oxygen substrate. Guanine is going to be oxidized, and they can produce the different molecule. For example, 8-oxo-7-8 dihydroguanine, 2,5-diamino-4-H imidazole 4N, and the third molecule is guanidine hydrodentoin and hydroguanidine hydrodentoin. So these are the 4 different molecules when the guanine is going to oxidize. So this way they are going to be use this base excision repair mechanism where the DNA glycosylase, they are going to break the beta and glycosidic bond, remove the damaged DNA from the base nucleotide. They can create an AP or a basic site. And what is the AP endonucleases is they can cleave the phosphodiester backbone immediately adjacent to the damage by generating the single-stranded break. And the AP exonuclease, they can completely remove the sugar phosphate. So deoxyribose phosphodiesterase, they can remove the few more nucleotide from the damaged strand. And there is the enzyme that is the DNA polymerase 1. They can synthesize the remove DNA with the proper and the correct bases. And once they reach and the gap, RNA is there, and that is going to fill with the DNA ligase that is called the sealing enzyme. So in this way, they can seal the gap. So this is the uh, more atomic level structure, how the AP endonuclease, they can catalyze the hydrolytic incision of the phosphodiester backbone immediately adjacent to the damage. They can generate the single stranded break with the 5-hydroxyribose phosphate and the 3-hydroxy end, and AP endonuclease, they can completely remove the phosphate backbone. So this is the way how they are going to correct the damaged DNA. There is the another mechanism for the DNA repair at the single stranded break level. So that is called the nucleotide excision repair. So in the nucleotide excision repair, they are going to be normally when the ultraviolet light expose the DNA, they are going to make the thymine dimer. So thymine dimer is normally help to block the DNA synthesis, and normally we use this UV light in the laminar airflow, where we are going laminar airflow, where we are going and culturing the bacteria, fungus, yeast, mammalian cell. So normally before starting the experiment, we use this we use the ultraviolet light to expose half an hour of the entire laminar air hood, where we feel there is the microorganism, and this microorganism, it is going to feel, it is going to kill with this UV light, because the UV light can create the thymine dimer in between the DNA, and the DNA is not going to replicate, and they can terminate the genome synthesis, and they can terminate the whole cell physiology. So there is the cytosine and the thymine, and this thymine, they are going to form the dimer, and once they are going to form the dimer, they can make the cyclobutane ring. So thymine dimer, they're going to form the cyclobutane ring in the adjacent thymine dimer. And this damaged DNA is due to the cross-link with the guanine nucleotide between the adjacent bases, where there is the nitrogen mustard and the DNA. They are going to make this N-guanylyl DNA monoadduct, and they are going to make this cross-link interstrand and the cross-link intrastrand. So in the interstrand, there is the complementary DNA, they are going to form between the one basis to another basis, so they can stop the activity of the DNA, so they can make the DNA damage. And another is the intrastrand, where the single strand, two bases, they are going to cross link, so they can also damage the DNA. So it can be repaired by the nucleotide excision, where number of the microbes in the environment normally encounter with the ultraviolet light, and they can cause the never-pyrimidine bases, thymine, cytosine, to form the dimer. So thymine dimer are the most common mechanism found normally in the environment, where the bacteria all the time they are going to expose. So pyrimidine dimer can be repaired by the several mechanisms, including the method that is called nucleotide excision repair. In this system, three subunit of the endonucleases, which is made by the UBA, B, C, and they can excise the passage of 12 to 13 nucleotide spanning in that dimer, and this is going to be ATP hydrolysis that can provide the energy for the many steps throughout the process. So ATP can help, and they can provide the energy in entire process of the DNA repair. So nucleotide excision repair, where the thymine dimer is going to form, and they can dimer the DNA. So these three enzymes, UBA, B, and the C, they are going to bind with the damaged DNA. So UBA, the function is they have the DNA binding protein and ATPase activity. So they can bind with the DNA and they have the ATPase activity. So a dimer DNA is recognized by the UBAB, two molecule of the UBA and one molecule of the UBB. It is going to bind with this DNA, and they can scan the DNA for the abnormalities, means where the damage is going to happen. Once they are going to recognize the DNA damage, then UVB is going to melt that location that the DNA, and the UBC have the endonuclease activity, so they can cleave the DNA. So the two molecule of the UBRA, it is going to bind in the DNA. So they are going to repair is going to start it. So they are going to begin with the association of the UBA two molecule and the UBB, and they hydrolyze by the ATP. So UBAB complex, it is going to bind to the DNA, and UBA makes the initial recognition of the damaged DNA. So once they are going to identify UBA, they bend the DNA near the damaged site. With the more ATP hydrolysis, UBA load the UBB onto the damaged DNA, after which UBA ejected or removed from the complex. So once the UBB is going to bind the damaged DNA, the UBRA it is going to remove, and the UBB cause the total melting of the DNA helix that can facilitate the insertion of the UBA beta hairpin between the DNA strand. So once the next step is UBB bind to the UBC, and UBC nick the damaged strand at the two sides, flanking the damaged bases. So the function of the DNA UBRC is the endonuclease. So UBRC both inside the 5' and the 3' of the lesion, and the N-terminal part 3' incision, C-terminal part 5' incision. So the another complex that is called the UBRD that had the helicase activity strip the resulting short nucleotide single stranded damage fragment from the DNA molecule. So UBRD helicase, they are going to help, and they have the DNA dependent ATPase activity. They can unwind the DNA duplex, and they can initiate unwinding more efficient from a nick substrate than the double stranded DNA complex. Once the single strand gap remain has the 3-hydroxyl group that allow DNA polymerase synthesis and the new DNA. After the DNA polymerase is finished, the ligase enzyme, it is going to seal the remaining gap in the DNA, and that's help to complete repairing of the damaged DNA. So this is the function and property, how they are going to remove and how they are going to help to repair the mechanism. So another single strand break, it is going to repair with the SOS response, where SOS response, they are going to damage, either they can enhance the cell death, and it is mediated by the radiation or genotoxic drugs mediated DNA damage. So in this two way, SOS response is going to help. And in the SOS, there is the SOS response regulator that is called the LAX-A dimer, which is transcriptional repression. LAC-A is activated following DNA damage. So in order to understand how they are going to regulate, so we need to understand first what is the LAC repressor. So the LAC operon was discovered by the Jacob and Monod in 1961, and they got the Nobel Prize for this important discovery in the science. And this is one of the breakthrough mechanisms they have identified in the history of the molecular biology. So the LAC-I is kinds of the transcription factor, and the transcription factor is a kinds of protein that can repress or activate the gene function. And this protein is expressed in the genome, and they are making the mRNA, and subsequently they are going to make the protein, and this protein is called the LAC-I protein. So LAC-I protein have the ability to bind the promoter region of the operator, and the operator is the small sequences which is found in the promoter, and they have ability where the transcription factor is going to bind. So promoter is the very unique sequences where the RNA polymerase is going to bind, and they can stop the transcription process. So once the LAC-I is active, they can bind with the operator region of the promoter, and they can stop, they can switch off the transcription of the subsequent gene like the LACZ. So if you use the lactose molecule that can bind with the LAC-I protein, and they can, this complex, it is not going to bind the operator region, and the operator region is free, then the RNA polymerase can bind with the promoter region, and they can express the LACZ. So it can start the expression of the LACZ. So this is the kinds of the system is on. So this system is regulate whether it's off or on. It depends upon the transcription factor, whether the transcription factor is active, inactive when they can bind with the lactose, IPTG like inducer molecule. Similar system it is going to happen in the SOS response where the LAX-A is the repressor protein, and they cannot allow the RNA polymerase can bind there. So there is the operator for the LAX-A, and they can normally produce in the system, and they can bind with the operator region. Once they are going to bind the operator region, they cannot allow the RNA polymerase can bind there, and if the RNA polymerase cannot bind there, the gene for the SOS repair is not going to active. So this is kinds of the regulatory mechanism where the SOS gene, it is not going to active, so the repairing mechanism cannot be possible. So this system, that is the RAC-A, which is going to activate the DNA damage, cause the LAX-A to undertake the autocatalytic cleavage, and they can inactivate the LAX-A repressor function that can turn or suppressed by the LAX-A. So in this mechanism, you can see the LAX-A, its kinds of the protein they are going to produce, and they are going to suppress the LAX-A gene. And in the same time, they can also suppress the target gene. That means the gene they are going to involve in the SOS repair. So once the RAC-A, it is going to induce, they are going to cleave the LAX-A. Once the LAX-A is going to cleave, they did not have ability to bind the promoter, and this SOS related gene, it is going to working, it is going to functional, it is going to be on. So in this way, when the gene is going to on, the repair system, it is going to activate it. So LAX-A normal condition, this is the small kinds of the protein, and the function as the as a dimer. So it is stable in the healthy cell, and it can repress the SOS box gene expression. And the LAX-A DNA binding, DNA damage condition, the LAX-A has silent proteolytic activity. And the RAC-A activates the autocleavage activity of the LAX-A dimer, and the DNA repair enzyme, it is going to be expressed. So this is the way how they are going to work. And in this SOS box and the LAX operon, you can clearly see that LAX-A dimer, they are going to bind with the operator region of the SOS box, and the upstream there is the promoter, and this SOS box, they have the different gene like the UBR, UMUC, UMUD. That's gene, it is going to help making the SOS response active. So once this dimer is going to clip, once the dimer is going to clip, the operator is free and or they are lax is going to degrade it, then operator is free and the RNA polymerase is going to bind on the promoter region and they can express the UBA, UMC and the D gene that's help in the DNA SOS response mechanism. So if the DNA it is going to damage and the lax A, they are going to make this lax A and this lax A it is going to clip. So there are the number of the gene that's help in the SOS repair mechanism. So this is the error-free DNA repair, that is the UBA, B, UBRC, UBRD, and they can transcribe UBABC endonuclease and they can initiate the NER error-free DNA repair system. There is another error-prone DNA repair, that is UMC, UMD and the UMDC. They are going to activate if UBRA remain inactive and they can produce the DNA polymerase 5 when the DNA damage occur at the multiple position. DNA polymerase 3 and the 1 is stalled, DNA polymerase take over the DNA replication, and that is the error prone DNA repair system. So the DNA repair, you can see normally once the RAC is activated, then the expression of SOS gene, once they are going to access, they can produce. When they are going to repress, then they are not going to produce. So RAC-A protein is the homologous recombination of the DNA. They can activate following the DNA damage. And they can recognize single-stranded DNA. They can inhibit the DNA replication. They can trigger the SOS response. So the SOS response, this key concept is the damage to the DNA caused by the A to trigger the SOS response. So RAC-A activates the auto cleavage activity of the LAX-A. LAX-A no longer can repress SOS box. LAX-A auto cleavage activate DNA repair enzyme based gene and they allow to be damage to be repaired. So there is the, you see there is the, in the normal condition, there is the no DNA damage, but when the cell distress, there is signal, there is a damage, they can induce the RAX-A by the protease function. And this both the domain, both the dimer, they are going to produce the LAX-A and they are going to bind with this LAX-A repressor. So this is the way how the DNA repair system it is going to happen. So it can initiate with the several DNA damage by the UV radiation, yeah, genotoxic drug. So that can occur within a few minutes of the damaging treatment. So they can coordinate function of the many DNA repair enzyme. RAC-A activate the autoclavage activity of the LAX-A. LAX-A no longer can repress the SOS box, they can activate the DNA repair enzyme. So after the DNA damage is repaired, the LAX-A protein lost the ability to destabilize the LAX-A. LAX-A protein rapidly accumulated and turn off the SOS gene. So then the DNA damage is going to happen in the both the strand, that is the double stranded break. It is going to happen, and this double stranded break is going to repair with the mechanism, with the pathways the system had, that is called the non-homologous interjoining pathways and the homology homologous recombination. So this is the two pathways, two basic pathways that can allow to when the double stranded break is happen in the DNA. So what happened? There is the exogenous agent, that is the radiation, genotoxic, chemicals, mechanical stress. It is going to happen and make the DNA damage. So the DNA is going to completely break both the strand, and once the both the strand is going to break, it is going to repair with the cellular mechanism. So there is the three way of they are going to repair. One is the homology directed repair, another is the non-homology indisjoining, and the third is the macrohomology mediated indisjoining. So in these three mechanisms, the damaged double stranded break it is going to repair. So if this DNA it is not going to repair, then there is the misrepair. So before the translocation and after the translocation, the chromosomal abnormalities is going to happen because some part of that gene it is going to translocate it, and there is the effect of the double stranded break where sometime it is unrepaired break. So in this way, the faulty or repaired DNA, chemo or radiotherapeutic regime, so it is not going to repair. So in this way, if it's not going to repair, the cell is going to be death, and that's lead to the apoptosis. So once the DNA it is going to damage, they make the double stranded break, and this double stranded break it is going to repair with this non-homology indisjoining, and in this way it is going to occur at any phase of the cell division where the damaged DNA it is going to repair. It does not rely on a template. It means either addition or deletion can be possible. So sometimes gene can restore the function, sometimes the gene cannot restore the function, and the functionality of the gene it is going to be out. So non-homologous indisjoining is the intrinsically mutagenic loss of the DNA strand, and this it is very fast step, and it will 30 minutes it is going to repair. So double stranded break it is going to also repair with the homology recombination mechanism where it is restricted to the S and G2 phase, and it's require the sister chromatid as a template, and it has high fidelity of the DNA repair mechanism. There is the no losses of the DNA strand. This process is quite slow, and it will take around 7 hours to make this DNA repair. So in the non-homologous indisjoining, they are going to repair the double-stranded DNA when they are going to expose and damage with the UV radiation, and this DNA is going to be ligate, and the ligate with the blunt end of the duplex DNA. No need for the template, a homologous region in the DNA for this type of the repairing. DNA structure cannot be completely retrieved sometimes because of the addition or deletion of the nucleotide inside the DNA once they are going to damage. So the mechanism is the step first, there is a Q protein that can recognize double stranded break DNA, and there is the endonuclease make the blunt end of the double stranded break DNA, and the polymerase is going to synthesize the DNA, and the ligase enzyme they can seal the gap of the blunt in the DNA. Subsequently, there is the deletion and amplification of the DNA sequences. So in this way, they are going to repair. So if you see, there is a more detailed mechanism where the key Q protein is going to form the heterodimer that can recognize, detect the damaged DNA by binding to the broken end, and the Q can bind and broken end together by binding to the two DNA molecule, and there is the another kinase that is the DNA PK. It is kinds of the kinase that can act as a molecular sensor for the DNA damage, and this DNA KP bind to the Q protein and the artemis, And this DNA KP is going to phosphorylate the Artemis, and Artemis is the kinds of the endonuclease. They can trim the DNA, and there are the certain unknown enzyme that is not uh, clearly known now, so they can also help the blunting of the broken DNA. And there is the enzyme that's LIG4 and XRCC4. They are going to ligate the blunt-ended duplex DNA. So the POO protein, they are going to have a heterodimer activity, and it's 70 and 80 POO, and they are recognize the double stranded break. And the crew have no conformational enzymatic function. Many have the helicase activity, ATPase activity, and that can improve the stability of the non-homologous end joining at the DNA endpoint. They can also carry their function for the longer period of the time. DNA PK is the serine threonine protein kinase. They can act as a molecular sensor for the degraded DNA, damaged DNA. They must be bound with the DNA to express the catalytic property. So in this mechanism, Artemis is it, so the endonuclease activity, they can activate the DNA KP bind to the Artemis, and they can activate the DNA KP, and they acquire the endonuclease activity. Artemis is remove the overhang, and the LIG4, this is the DNA ligase enzyme. They can also XRCCR4 X-ray repair cross complementary protein, and they are going to produce the blunt in DNA strand that is going to bind to the LIG4, and the LIG4 XRCC4 complex is responsible for non-homologous end joining ligase step, and CXCCR that can enhance the joining activity of the LIG4. So it can help to make the better ligation activity. And there is another factor called the XLF. So XLF is the XRCC4 like factor. They may serve as a bridge between the XRCC4 and the other non-homologous end joining factor located at the end of the DNA. So end product is the DNA is going to restore their integrity, and sometime it can lose the DNA, it is going to be occur. So there is the another mechanism where the DNA damage is going to repair, that is called the homology recombination mechanism. So in the homology recombination, where the double stranded break, it is going to repair with this mechanism. So homologous recombination, it is restricted with the S and the G2 phase, and it's required the sister chromatin as a template for repairing of the double stranded break DNA. It is very high fidelity repair mechanism, means it is dominantly present in the cellular system, and no loss of the DNA strand because they have the homologous region and they are going to easily combine together. And this process is quite slow, and it is taking around 7 hours to complete this process as compared to the non-homologous end joining. So in the non-homologous end joining, it's take only the 30 minutes. So the homologous recombination mechanism, it's quite long time required, and it is slow process. So normally the double stranded break is going to happen with the ionizing radiation or the genotoxic chemicals. There is the highly cytotoxic DNA damage, and they can disrupt the genomic integrity because it is going to completely disrupt the function. And they are going to repair with the formation of the single stranded DNA tail. The broken end is the process to form a 3 single stranded tail, and that is 3 end can be extended. So the stand inversion is going to happen by the repairing of the 3 in single-stranded tail with the appropriate segment of the sister chromatin. So homologous recombination, it is going to happen with the DNA repair synthesis without loss of the single nucleotide, because the another end have the homologous region, and with the ligase enzyme they can seal the gap once they are going to find the homologous region. So if you go more detail about the DNA double stranded break, it is going to repair by the homologous recombination mechanism. So they are going to first recruit the MRE11 factors, and that contain the endonuclease exonuclease activity, and they are going to produce the single stranded endonuclease activity and the double stranded specific 3 to 5 exonuclease activity, and they are found in every kingdom of the life, and they require RAD50 and the ATP molecule for the recombination is going to happen. After that, they can recruit the RAD50 protein, which is the ATPase, and they function as a dimer, and also the DNA repair protein, that is the bind with DNA in and hold them in a close proximity, so they are going to putting together and the closely found, and they can found also in the entire kingdom of the life. Once the it is going to bind there, then there is the recruitment of the NBS1. They can stimulate the endonuclease activity of MER11, and this is the kinds of the linker protein. They can recruit many other protein enzyme that's involved in the DNA repair pathways, and which is less conserved, and the yeast, this protein is called the XRS2. Once the third step is completed, they can form the complex. They can again recruit the CTIP, that is the CTIBP interacting protein, and which is also called the retinoblastoma 1 binding protein 8, and in the yeast this protein is called SEA2, and it is kinds of the endonuclease. They can cooperate with the MER11, lambda 50, and the NBN complex. So this complex, it is going to initiate the DNA insection. And there is another enzyme that is called the XO1 endonuclease. This XO1 endonuclease, it is going to make the 5 to 3 exodeoxyribonuclease activity, and they have the extensive resection carried out by the XO1 and MR1 complex. They can be detached. So there is the replication protein A that can bind with the single-stranded DNA, and they can coat the 3 hang in with the single-stranded overhanging immediately. So this homologous recombination, it is going to happen in the in the meiosis in order to make the double-stranded break repair. Once the recruitment of the RPA lambda 51 BRCA, the replication protein A that is going to bind with the single-stranded DNA in eukaryotic cell, and they can coat 3 in the single-stranded overhang immediately. And another protein, that is the lambda 51, they can replace the RPA. They can catalyze the recognition of the homologous strand and stand between the homologous and the DNA partner, and they can form joining between this process DNA break and the repair template. So this is the BRCA2, BRCA1, that is the breast cancer type 1 and 2 susceptibility protein that's enable the lambda 51 to displace the RPA to form the single-stranded DNA. And the lambda 51 and the lambda 54, they can promote and replacement of the RPA and the processing of the DNA strand that contain the 3 overhang. So lambda 51 and the lambda 54, they are going to make the strand transversion. DNA polymerase, they can start the DNA synthesis and they can extend the broken end strand. And the lambda 51 and lambda 54, they are going to make the heteroduplex complex and the branch migration. And the DNA polymerase, they can synthesize the DNA and resolvase and the topo. This is the enzyme. They can help to make the resolution of the poly-D junction and the formation of the crossover. With the help of the ligase enzyme, both the strand are going to seal and they can repair the complete pathways. Once they are going to seal, then we can see what are the differences among the E. coli, Saccharomyces cerevisiae and the human, where the homologous recombination it is going to happen. So the initiation factor for the homologous recombination in E. coli is the RAG-BCD, and the Saccharomyces cerevisiae, the initiation complex protein is the MER11, RAD50 and XRS2. And in the human, the initiation for the homologous is the MER11, Lambda50 and RAD50 and the NBS1. Homologous repair and the DNA strand exchange, it is going to happen with the RAG-A and the single stranded binding protein, SSB. In the Saccharomyces cerevisiae, lambda 51, DMC1, RPA, lambda 52, lambda 54, these are the protein they are going to involve for the homologous repairing. In the human, lambda 51, DMC1, RPA, lambda 52 and lambda 54, these are the same enzyme they are going to involve in the human and the Saccharomyces cerevisiae. And the formation of the DNA heteroduplex that is going to be extend in the E. coli RUVAB, the enzyme found in the E. coli for making the heteroduplex extension. In the Saccharomyces cerevisiae and the human, this is the same protein RAD54 involved in the heteroduplex extension. Resolution and dissolution which is going to happen in the E. coli, that's protein, this enzyme called the RUVC and the topo3. Topo3 is the same in the Saccharomyces cerevisiae and the human also. And there is also the another protein resolvase A, which is found in the human. So the last mechanism for the DNA repair on the single stranded break is the direct repair by the methyl transferase and alpha ketoglutarate deoxygenase enzyme. So in this way, the adenine, guanine, thymine, and cytosine, these are the nucleotide which is going to be added, some of the methylated side, some of the O-methylated side, N-methylated side, and the O-methylated side. So position is vary from the adenine, guanine, thymine, and cytosine. So adenine and cytosine, when they are going to N-methylated, they are going to repair by the alpha keto dependent deoxygenase enzyme that is called ALK-B. ALK-B enzyme, when the adenine and cytosine is going to methylated, they are going to be repaired. Guanine and thymine, when they are going to O-methylated, then that is going to repair by the N-methylase transferase enzyme. So there are the number of the drugs molecule, which is anti-cancer drugs, alkylating agent, cyclosporamide, and melphalan and dicarbazine. These are the anti-cancer drugs that are going to be make the different changes in the DNA, and also there are the certain mutagen. They are going to change the basis. So the one is the EMS, that is the ethyl methyl sulfonate. MNNG, that is N-methyl N-nitro N-nitrosaguanidine, methyl methane sulfonate. So these are the mutagen. They are going to change the different a function group in this adenine, guanine, thymine, and cytosine. So when the adenine is going to expose with the EMS, DMS, and MMS, this is called the mutagen. They are going to make the 1-methyl adenine. So in 1 position and 3 position, it is going to be methylated. And this methylated DNA, it is going to be repaired with the help of the alpha ketoglutarate dependent deoxygenase, this ALK-B enzyme. This enzyme, it is going to act on 1-methyl adenine with the help of the alpha ketoglutarate, and they are going to make the formaldehyde, and H2O was removed, and also the adenine is going to produce. So this is going to repair by the alkylation of the DNA. They can remove the methyl group from the 1-methylene adenine, and the oxidation of the methylation that's required for the molecular oxygen and alpha ketoglutarate and the iron ion. So they can provide the extension of the resistance to the alkylating agent MMS and the DMS molecule. When the cytosine is going to expose with the mutagen like EMS and the DMS or MMS, they are going, 3' is going to methylated with this cytosine. And this 3' methylated cytosine, it is going to be remove or repair by the alpha ketoglutarate dependent deoxygenase enzyme, and it is going to repair by the alkylated DNA or RNA molecule. They can remove the methyl group from the 3-methyl cytosine, and also they are going to oxidize demethylation that's require the oxygen and alpha ketoglutarate as well as the ion, and they can provide the so this 3-methyl cytosine is going to be repaired by the removal of the methyl group from the 3-methyl cytosine molecule. So there is the oxidation of the methyl demethylation that's require the oxygen, alpha ketoglutarate, and the iron molecule that can provide the extension resistance to the alkylating agent. And there is another mutagen that is EMS, MNN, and MM, and the EMS. They are going to easily make the guanine, making this 6-methyl guanine. And once they expose the thymine, they can make this oxygen methyl thymine molecule. And this molecule, it is going to repair by this direct repair by the methyl transferase enzyme. So methyl transferase enzyme, they are going to make this unmethylated with this guanine. It is going to make conversion and they can be repaired. So this methylated their structure, so they are going to methyl transferase enzyme. They can help and they can remove the methyl group from the guanine and the thymine molecule in order to properly come back with the normal adenine, guanine, thymine, cytosine. So all the repair, it is going to happen. So this is the way how the DNA repair it is going to completed. So thank you so much.